Thank you very much for that introduction. Today's presentation is a paean to my PhD advisor, Dr. Richard Lepke, who had tried many times to sensitize FDA and other regulatory agencies about nitrosamines in pharmaceuticals. With that, I will directly go to my topic for today. Nitrosamines are a family of carcinogens which are formed by the reaction of secondary and tertiary amines and some other functional groups with nitrogen in the +3 state, especially nitrite. They are a cohort of concern and also possible and probable carcinogen according to IARC. Nitrosamines are all around us. Here are some sources of nitrosamines. But today we are going to specifically focus on the nitrosamine impurities in pharmaceuticals. Nitrosamines can be present as impurities based on the drug substance as well as the components of the drug product. And I have provided some sources here. Now, when it comes to drug substance, we have enough understanding of organic chemistry to apprehend where the nitrosamine could be formed and even control it sometimes. But that can't be said about the drug product. Especially the big unknown in the drug product are the excipients. Excipients can be as high as 90% of a formulation. They are manufactured under non-GMP condition, and most of the excipient manufacturers are not testing them for nitrate, nitrite, or small amines. Several drug substances are secondary tertiary amines which are capable of forming their corresponding nitrosamines when there is a nitrosating agent. I did a high-level review of just the drugs with monographs in USP, and this is my finding. While I will not go into details of this, I wanted to point out this highlighted information. Secondary amines, I found almost 95. Now, please understand that secondary amines can easily nitrosate and form nitrosamines if there are nitrites and nitrates present. And yes, nitrates, because nitrates can reduce to nitrite in presence of reducing agent, impurities and excipients and trace metals. So there may be two ways of getting nitrosamine-free drugs. Either we cut down the amines or we cut down nitrites and other nitrosating agents. What we just saw is that it's impossible to cut down amines because amines can be common reagents in drug substance manufacturing, but more importantly, a large percentage of the drugs approved contain amines as drug substance, secondary amines, tertiary amines, amines that can form dimethyl nitrosamine, diethyl nitrosamine, and so many others. So the best way of preventing nitrosamines in drugs may be to cut down the source of nitrate and nitrite. So we think that it may be worth exploring the feasibility of adding a trapping agent capable of scavenging nitrosating agents. I have given a few possible scavengers, but please don't think that you can just throw a pinch of any one of these and voila, your nitrosamines disappear. That's not going to happen because these are very, sensitive and they work at particular pH. They may be, not working the way they should in presence of certain excipients. There's a lot of research that needs to be done. And yes, again, as I said, there's a lot of research that needs to be done. Not much work has been done on nitrosamine since 1990 because of lack of grants in this area. People call nitrosamines a dry well. So how can FDA help? FDA can support the research related to prevention of nitrosation, maybe divert some of the GDUFA money in doing this research because generic industry is really struggling with the nitrosamine issue. The best way to mitigate the risk of nitrosamine is prevention of these impurities from being formed rather than trying to tighten their limits to impractical levels once they're already found. As they say, an ounce of prevention is worth a pound of cure. With that, I finish my talk. Thank you, and hope we can overcome this issue like we have overcome so many other issues in the past. Thank you.
Thank you. My presentation is regarding developing processes for nitrite-free excipients. Per ICHM-7, nitrosamines are in the cohort of concern. They're mutagenic carcinogens in animals and possibly in humans. FDA's February 2021 guidance for industry on control of nitrosamine impurities in human drugs requires nitrosamine risk assessments and mitigation of risk. The guidance lists 7 common nitrosamine impurities and recommends acceptable limits for 6. FDA and USP have also published analytical methods for these common nitrosamines. FDA acknowledges that there are other nitrosamines which can be formed based on the API manufacturing process or structure and recommends that these be controlled at 26.5 nanograms per day. Nitrites have been identified as the major source of nitrosamine risk, as they are common in many excipients and are instrumental in nitrosamine formation. Nitrosamines are formed when secondary and tertiary amines react with nitrites or other nitrosating agents. While quaternary amines are generally not known to nitrosate easily, they may have secondary and tertiary amines as impurities which can then form nitrosamines. Any source of nitrite or nitrate which can reduce the nitrite can increase the risk of nitrosamines in formulations. Drug product formulations consist of one or more APIs and excipients. Now, APIs are manufactured under cGMP conditions. Excipients, on the other hand, which may be the major constituents of a formulation, are predominantly manufactured under non-cGMP conditions. The pharmaceutical industry demand for excipients constitutes only a small fraction of the market for excipient manufacturers, which makes it difficult for them to meet the nitrosamine requirements. Several excipients are known to have residual nitrites and nitrates. Examples are like microcrystalline cellulose, pre-gelatinized starch, sodium starch glycolate, and povidone. Controlling nitrites and nitrates in excipients is challenging because detecting and quantitating at parts per billion levels in the formulation matrix is difficult. There might be interference from the soluble and insoluble components. The global nature of excipient manufacturing also makes it difficult to understand excipient quality. APIs, which are themselves secondary amines, may form the corresponding nitrosamines during manufacturing and storage due to reaction with nitrites in excipients. Amine impurities in APIs may also react with nitrites originating from excipients. In rare instances, excipients may have amine residues which could react with the nitrite residues in other excipients in the formulation to form nitrosamines. As excipients generally constitute the major portion of a drug formulation, not addressing the risk of nitrosamines arising from nitrites in excipients may make it difficult to remediate the risk when the API is a secondary amine, or when there are impurities in the API or the excipients which are themselves secondary amines. It may necessitate reformulation of drug products and or the conduct of expensive in vivo studies to justify nitrosamine limits. It may result in the withdrawal of drug applications or deter or impede development of critical much-needed medications. It may delay market introduction of affordable generic drugs and result in drug shortages. All of the above may impact the availability of cheaper generic drugs to the American public. We can remediate the risk of nitrosamines by controlling the nitrite level in excipients, preferably at parts per billion levels, to reduce the risk of nitrosamine impurities in drug products. We can identify the root cause of nitrites in excipients and or develop commercial processes for manufacturing nitrite-free excipients. Excipient manufacturers may not have the necessary resources to develop processes. There is undue burden on drug product manufacturers to evaluate the presence of nitrites in every lot of excipient. Now, how can FDA help? FDA can support a collaborative effort to understand the source of nitrites in excipients and propose improvements in the manufacturing process of excipients, leading to desirable nitrite levels. Thank you.
Good morning, afternoon, or evening, depending on where you are listening from in the world. My name is Martin Ehlert, and I'm speaking today on behalf of Apotex. Thank you for the opportunity to raise a topic that we at Apotex consider very germane to the current GDSR program. N-nitrosamines in drug products is now a widespread issue relevant to generic and brand products alike. Starting in 2018, there have been numerous recalls of products from U.S. pharmacies due to the presence of unacceptably high levels of nitrosamines such as NDMA and several other specific N-nitrosamines. The challenge with nitrosamines detection, quantification, and control as potential contaminants in drug products is burdened by a particular factor relative to other impurities in drug products. N-nitrosamines' current permissible limits are extremely low, with corresponding specifications often being in the hundreds or tens of parts per billion. Detection and accurate quantification at these levels is within the capabilities of current analytical technology, but it is definitely near the edge of such performance. For this reason, we advocate that FDA include in the science and research program funding to study the prevalence and sources of artifacts in trace nitrosamines analytical methods for drug products. Why does this matter? Well, there have already been at least 2 instances in the past 2 years where the agency has had to take issue with a company, namely Valisure, that made public erroneously high levels of NDMA in ranitidine and metformin drug products on the market. In both cases, the FDA had to clarify that some of the data Valisure presented either grossly or significantly overstated levels of NDMA in some drug products. In fact, in the case of metformin products, in July 2020, in the AAPS journal, Yang Marzan et al. From the agency's laboratories published an excellent paper warning how subtle errors in Valishure's LCMS method led to NDMA results that were substantially overstated in many ANDA-holders' products. I realize that a criticism of this point could be that poor method development on the part of one party is not a good reason to direct GDSR funds towards drug product research when there are so many other worthy topics. So allow me a minute to make the case that an insidious problem might be lurking behind the analysis of nitrosamines in some drug products. To reiterate, N-nitrosamines have very low acceptable intakes. The most facile path for N-nitrosamines formation is from the reaction of amines, particularly secondary amines, and nitrosating agents, which typically form from nitrite ion. The level of amines and nitrite precursors in drug products are frequently orders of magnitude higher than what would be necessary to generate corresponding nitrosamines at greater than acceptable intake values. Chemically speaking, none of the precursors are limiting reactants. Accepting this latter point, what regulators and industry currently have limited understanding about is when and how much of a given nitrosamine forms in the overall drug product manufacturing, release testing, and stability testing process. I highlighted the word testing on the previous slide, and here is the crux of the matter. Nitrosamines limits for drug products are so low that the only analytical tool we have in our toolbox is chromatographic methods coupled with advanced mass spectrometric detection. There aren't truly orthogonal direct analysis methods available to cross-check GC or LCMS methods with the sensitivity needed for nitrosamines. All of these GC or LCMS methods require dissolution-based sample preparation with contact times spanning from minutes up to about an hour to extract the potential nitrosamines for detection and accurate quantification. And they assume the almost subconscious tenet. The analyte is already present in the test article. How does one know whether this holds true for ppb levels of nitrosamines for which the reaction precursors are known to be present at high levels in the test article, namely the drug product? Current good method development and validation guards against scenario B, shown here, and ensures that methods work per scenario A. But these same techniques and practices are unable to differentiate between scenario C and scenario A. We believe that funding research into the prevalence and severity of scenario C for nitrosamines in drug products would be money well spent and could help to avoid some unnecessary recalls or product withdrawals, which is to the benefit of patients, the agency, and industry alike. Thank you again for providing this time and for your attention.
Thank you for your kind introduction. I want to make a public comment on creating a pathway for regulatory approval utilizing a waiver for BE studies conducted with the US RLD based on BE studies conducted using non-US RLD and in vitro characterization similarity as bridging. I have a disclaimer that the opinions expressed herein are solely those of the presenter and industry collaborators and not represent the statements or opinions specific to Sandoz Inc. or Novartis Pharmaceuticals. This comment was put together in collaboration with colleagues from Sandoz, Actavis, and Apotex listed below. Thanks to Lisa Parks of AAM and FDA Generic Drug Science and Research staff for providing guidance during this process. My comment covers to the concerns that the generic industry and patients have, and also a solution-oriented proposal, and finally a request to the GDUFA research team. There is a real challenge to the generic entry worldwide, including US, due to patient and financial constraint to develop generic drugs that require PKBE studies and CEBE studies in patient populations for registration. Each region now requires in vivo BE study with a regional R&D that is a heavy financial strain on generic industry that reflect expensive, lengthy studies with basically same R&D. It is an unnecessary duplication and drug exposure with no real patient benefit and results in higher healthcare costs due to lack of generic drug availability. In the past, FDA has granted waivers of PK studies in patients with clinical endpoint studies for generic complex products with Q1, Q2, and characterization similarity data. A similar biowaiver concept can be extended to certain qualified products that require PKBE or CEBE studies in patients if additional BE data utilizing the non-US RLD as a comparator is also submitted. Our proposal is to allow the option of waiving BE studies conducted with the US RLD based on BE studies conducted using non-US RLD if it meets the following criteria. In vitro characterization similarity of test product is established to the US RLD and also non-US RLD using the same criteria already described in the US product specific guidance. In addition, PKBE or CEBE was established as per US product specific guidance except non-US RLD from a major ICH region was used as a comparator. We feel there are some products to consider for this approach, such as long-term injectables, long-acting products, or locally acting products, including inhalation products that require CEBE studies, and also drug products with high clinical burden like oncology and antipsychotic products. Our request to the FDA research team is to allocate resources to explore possibility of providing an optional biowaiver pathway in the product-specific guidance for products with high clinical burden, utilizing data from PKBE and CEBE studies conducted using the test versus non-US RLD, and also in vitro characterization similarity between test and non-US RLD using the same criteria described for test versus US RLD in the PSG. We want FDA to explore implementing this innovative regulatory biowaiver pathway under 3FR 320.22 without requiring legislative procedures. We also request FDA to discuss this approach with ICH partners for harmonization and common understanding for mutual utilization of BE data as much as possible for products that require high clinical burden to register. In this context, we would like to make a reference to an earlier talk given by Kiran Krishnan of Apotex for similar topics expressing similar concern. In summary, we sincerely hope that OGD can establish a bioaware pathway that we propose to help the generic industry bring some more affordable, quality generic medicines to the US market that now require expensive clinical trials for registration. Thank you for your attention.